biojourney. In last video we talked about the structure of bisexual flower and stamen. Now as we know that for sexual reproduction there must be the presence of male gamete and female gametes. So in this video we will be discussing about the internal structure of anther of the stamen. So as we know, anther is the most important portion of the stamen which is responsible for the formation of microspore or pollen grain, which is the representative of the male gametes. Okay, so let's see what is there inside the anther. So let me draw first the structure of stamen that we discussed in the last video. Now this is the stamen. Okay, if we take the TS of the anther, we can see in this bilobed structure with two theca. Bilobed structure with two theca means total you can see over here 4 different regions we can see in the TS of the anther. Let me write here bilobed structure with two theca. This is known as ditheca structure. So two major portion further having two sub portion. So here you can see these 4 different regions of the anther. So anther is a 4-sided tetragonal structure. Now this each region that is known as microsporangium. So total 4 are there. That's why it is known as microsporangia. Okay, microsporangia. How many? 4 are there, right? Now internally, if we see in the center of this TS of the anther, here the sterile tissue portion is there, that is known as connective. This is the sterile region which is not involved into the formation of the pollen grain. It provides only the connection to the filament. Now these 4 regions are the important region for the pollen grain formation. In the center, if we see the collection of cells, that is sporogenous tissue mass that is present, okay, here, and it is covered with the 4 different layers. Here now you can see this 1, 2, 3 and 4. These 4 different layers are there and they are protecting and providing nourishment to the inner cell mass. This is inner cell mass of each microsporangium. So this type of same structure you can see over here also, because this layer is in continuation like this. Okay, same here also. Now if we enlarge this structure, microsporangium, we can see this type of wall layers of the anther of the microsporangium. Okay, so we'll see the name of this all. Outermost protective layer of the microsporangium of the anther, that is known as epidermis. This is first one from outside in, if you see. Second layer, that is known as endothecium. Then these 2 you can see blue color structure, blue color cell layers, they are known as middle layers. Okay, second and third. And this pink layer, that is known as tapetum, 4th one, which is directly having the connection with this inner cell mass. This inner cell mass only is the important structure which is known as sporogenous tissue. Okay, so here you can see these 4 different layers and this inner cell mass. Now, how this pollen grain that will germinate, okay, in this sporogenous tissue, such a process is known as microsporogenesis. Microsporogenesis means the formation of microspores from this sporogenous tissue. So here you can see many cells are compactly arranged in this center region. If we take one cell, so that is PMC, means pollen mother cell. Only one cell here I am taking, okay. One cell that undergoes the process known as meiosis. Now this meiosis and mitosis process you studied in the 11th standard, okay. Meiosis, because of the meiosis process, this pollen mother cell, which is having the genetic material twice n, that turns into, means reduction of the chromosomal material turns into the n number of the genetic material. Lastly, after the successive meiotic division, this PMC turns into tetrad. Okay, means 4 cells. You can see these 4 cells are known as microspores. These microspores that turns into the mature condition, they are known as pollen grain. And each pollen grain is a representative of male gametophyte. Okay, so this is the important cell for the reproduction, okay, from the male side. This whole process here we have discussed, that is microsporogenesis. Okay, under the banner of meiosis, PMC that turns into tetrad, that is the formation of the microspores over here, that turns into pollen grain, and pollen grain that is representative of the male gametophytes. Okay, now after the completion of the whole process, here you can see this portion, it is full, it will be full of pollen grain. Okay, and slowly, gradually, what will happen here, these two layers, they will provide the nutrition to this developing inner cell mass, and at last in a mature anther we can see only these 2 layers and this inner cell mass that turned into pollen grains. So there are lots of pollen grains after the completion of whole this microsporogenesis in each sporangium. So now you can imagine 4 microsporangia are there. In these 4 microsporangia, each is having this cell mass that undergoes the process of microsporogenesis. So lots of pollen grains that will arise from this inner region. Okay, and that will come out with the help of endothecium to the outer surface of the anther. So in flower you can see sometimes the yellow powdery material that is coming out, that is here this type of structure you can see, that is the matured pollen grain representative of the male gametophyte. Okay, now in next video we will see the detail of the pollen grain. Okay, the outer structure, inner structure, and the germination inside what is there, that we will see in the next video. Okay, let's see the NCERT plus content now. Here we have discussed the 4 different layers of the anther. The outermost, that is epidermis. Okay, if you see a little bit more about the epidermis, it is a unilayer, means single layer is there. And what is the nature of this epidermis? It is protective. It provides protection to the inner material. Second one, that is endothecium. Okay, in endothecium, it is also single layer. Radially elongated cells are there, and fibrous thickening of alpha cellulose we can see, means the deposition of the alpha cellulose we can see in the endothecium. So this is the important to remember. In some anther we can see the deposition of pectin and lignin also. What is the function? Endothecium helps in the dispersal of pollen. So after the completion of whole process of the microsporogenesis, inside the microspores that turns into the mature pollen grain, that mature pollen grain that will come out from this. So the bursting of this layer that leads to the dispersal of the pollen grain, and all the pollen grain we can see over the outer surface of anther. Then after third layer, middle layer, number of layers we can see 1 to 6. Now in our diagram we have shown 2 layers. Okay, cells are flattened and crushed, made up of parenchyma tissue. What is the function? It stores the reserve food, means it supplies the nutritive material to the developing sporogenous tissue. Then the innermost layer directly attached with the sporogenous tissue, that is tapetum. Single innermost layer with viscous and dense cytoplasm with prominent big nucleus or with many nuclei. Okay, according to the behavior of tapetum, they are of 2 types, amoeboid and secretory. Okay, it is also nutritive. And one more thing over here you can highlight, UV bodies are present into the tapetum which secrete the most important material that is known as spore pollenin. And the spore pollenin is an important material for the exine of the pollen grain. Okay, so here these 4 layers are there of the anther. Now if we see the development of anther, little bit we will discuss here. Development of anther. Okay, initially anther is the homogeneous mass of, it is homogeneous mass of meristem. Okay, meristematic tissue. Then after it turns into 4-lobed structure. Structure of archisporangial cells. Okay, archisporangial cells further modified into 2 types of cells. One is known as primary parietal cells, and another that is sporogenous cell. Okay, this primary parietal cell that turns into, means that divides several times, and that will make the walls of anther. And here this also divides few times, and that will make microspores tissue. Okay, in which all the cells are known as PMC. Okay, thank you.